Okay, good morning students. In previous class, under the chapter cell cycle and cell division, we had discussed regarding the what meiosis, the definition of meiosis, and then we had discussed about the particular events which are going to take place in a meiosis, that is the karyokinesis and cytokinesis, as same we are studying in mitosis. And afterwards, different phases of meiosis which are going to complete in meiosis 1 and meiosis 2, I told you. And in meiosis 1, again there are the four phases: prophase 1, metaphase 1, anaphase 1, telophase 1. And meiosis 2, we are going to study prophase 2, metaphase 2, anaphase 2 and telophase 2. Okay. So in today's class, we are going to start discussing about the meiosis 1, and in meiosis 1 we are going to discuss the prophase 1. In the sense, meiosis 1 consists of prophase 1, metaphase 1, anaphase 1 and telophase 1. Okay, so first the prophase 1 of the meiosis 1, it is a longer and complex when compared to the prophase of a metaphase. When we are going to compare with the prophase of a metaphase, where we are studying metaphase, it is equational division and the two cells are going to form, and there we are studying the phase, prophase. That is, when we are going to compare with that phase, the meiosis 1 of the prophase 1 is longer and complex when compared to the prophase of the metaphase. And what is going to happen here? This prophase is again divided into 5 phases. This is very important. Sometimes the question is going to ask, explain what is meiosis. What is meiosis? Explain the phases of the prophase 1. Here, the prophase 1 is a longer and complex when compared to the prophase of the mitosis. And we are not studying further phases in a what prophase in mitosis. But here in meiosis 1, prophase 1, it is again divided into 5, what we are going to call them as a 5 sub phases. What are those? In the sense, one is leptotin, zygotin, packaging, diplotin and dikinesis. And here one mass question bala. Okay, separate one mass question kelbodu. They used to ask you one mass question. In the sense, what is going to happen in a leptotin, what is going to happen in a zygotin, what is happening in a packaging, diplotin, dikinesis. Now we are going to study one by one. 5 mass question here which explain all these things. Now leptotin, prophase 1 of the leptotin, you all know about the prophase. Prophase like in prophase, the chromatin is start condensing. It is going to go for formation of chromosome, nuclear envelope. Okay, it is going to disappear at the culture. Likewise, only here also those processes are going to go, but some complex processes we are going to study in a prophase 1. In that first phase, it is a leptotin. Leptotin, here the chromosomes become gradually visible under the microscope. Okay, and the compaction of the chromosome. In the leptotin, what is going to happen? The chromosomes are going to visible under the microscope. In the sense, chromatid are going to start condense and they are going to form a chromosome. And compaction of the chromosome ಅಂತಂದ್ರೆ ಏನ condensation ಅಥವಾ formation, compaction of the chromosome is continue throughout the leptotin. ಅಂದ್ರೆ ಏನ, in leptotin phase you are going to see the chromosomes under the microscope and the compaction of chromosome throughout the leptotin. Chromosome throughout the leptotin. Understood? Then next, after this, leptotin is followed by the zygotin. What is going to happen in a zygotin? In the sense, whatever the chromosomes are found in a leptotin, they are start pairing. In the sense, how? Suppose this is one chromosome and this is another chromosome. These are start pairing like this. Our samay barakiya. Zygotin. Chromosomes start pairing together and the process These are called as a homologous chromosomes. And chromosome synapses is accompanied by the formation of complex structure. It is going to form a complex structure called as synaptonal means synaptonemal complex. Synaptonemal complex. Synaptonemal complex. Synaptonemal complex is a pair of synapse homologous chromosomes called as bivalent or a triplete. Bivalent or a tetrad. Bivalent or a tetrad. When they are going to pair, and that paired structure is called as a synaptonemal complex, and that is what we are going to call as a bivalent. Bivalent or a tetrad. That is happen in zygote. Zygote leptotin chromosomes are gradually visible under the microscope. Compaction of the chromosome taking place throughout the leptotin. Then leptotin is entered into the zygote. In zygote, whatever the chromosomes are formed in a leptotin, those are start pairing together. Coming to each other, and they are going to form a pair. And that pair, that process is called as a synapsis. And the pair chromosomes are called as a homologous chromosomes, and they are going to form a structure. That structure is called as a synaptonemal complex. We are going to call them as a bivalent. It is the type of that is. In a packeting, what is going to happen? Packeting those are going to start pairing each other like this. This is a pair right here. This one the pair now pairing each other, crossing each other like this. This is the crossing. Okay, I hope you are getting my point. ಈ ಪ್ಯಾಕೆಟಿಂಗ್ ಬಂದಾಗ ಇವೇನು ಕ್ರೋಮೋಸೋಮ್ಸ್ ಪೇರ್ ಆಯಿತು those crossing over occurs. ಇಲ್ಲೇನಾಗ್ತೈತಿ ಪ್ಯಾಕೆಟಿಂಗ್ನಾಗ crossing over occurs between the non sister chromatids. Chromatids ಅಂದ್ರೆ ಬೇರೆ ಮೀನಿಂಗ್ ಇವು ಮ್ಯಾಗ ಬರ್ತಾವೆ chromatids ಹೌದಾ. ಈ non sister chromatids ಏನಾಗ್ತೈತಿ ಅಂದಾಗ homologous chromosome ಇವೆಲ್ಲ ಕಂಪ್ಲೀಟ್ homologous chromosome ಅಂತ non sister chromatids ಏನಾಗ್ತೈತಿ exchange what genetic material. Crossing over exchange the genetic material between the homologous chromosomes. ಇಲ್ಲಿ pair ಆಗಿ ಏನಂದ್ರು chromosome appear ಅಂದ್ರೆ ಏನ ಈ chromosome ಕಾಣಿಸಕತ್ತು under the electron microscope. Then these are paired here. Afterward packeting ನಾಗ ಏನಾಗ್ತೈತಿ ಅಂದಾಗ crossing over ಆಗ್ಬಿಟ್ಟು crossing over ಆದಾಗ ಏನಾಗ್ತೈತಿ ಒಂದು chromosome ನ genetic material ಇನ್ನೊಂದು chromosome ನ genetic material ಏನಾಗ್ತಿತ್ತು exchange ಆಗಿತ್ತು. That is what crossing over exchange the genetic material between the two homologous chromosomes. And this crossing over it is mediated by some enzymes. That is very important. That is also very important. Crossing over it is mediated by the enzymes, and those enzymes are what called as a recombinase. One last question here. Whether the packeting or a crossing over it is a enzyme based process? Yeah, it is enzyme related process of that, and the enzyme is involved in a crossing over that is called as a recombinase. Okay, then what is going to happen? Again ಒಂದು chromosome the genetic material start exchanging, and that is what we are going to call as a recombination. Recombination of genetic material. Again recombination of genetic material. Genetic material recombination and that is the end of packeting. This is packeting. Then diplotin. What is going to happen in a diplotin? Even crossing over those again they are going to start separating. In the sense, homologous chromosomes separate to each other except the site of cross. Where they are crossed, a cross of the jaga bittu baki place there they are going to separate with each other. These X-shaped structures, X-shaped structure are like this. This is X-shaped structure, and that X-shaped structure, see, is X-shaped. In the apart from this place, they are going to start separating in a protein, and that is what chiasmata. That is what chiasmata. And this diplotin is to end 1 month ಅಥವಾ 1 year ತನಕ ಈ diplotin ಇರ್ತೈತಿ. ಎಲ್ಪ ಅಂದಾಗ oocytes of some vertebrates, oocytes of some vertebrates, in oocytes of some vertebrates, the diplotin, it is like that only. It is not going to enter in a dikinesis. How many time means how many days? 1 month or up to 1 year. 1 year ಅಥವಾ 1 month ತನಕ ಏನಾಗ್ತೈತಿ ಅಂದ್ರೆ diplotin ಅಂತ ಉಳ್ಕೊತೈತಿ before going for the dikinesis. So diplotin ದಾಗ ಏನಾಗ್ತೈತಿ chiasmata. Chiasmata ಅಂದ್ರೆ ಏನ? genetic recombination ಆಗ ನಾವು chromosome separate ಆಗ್ತಾವು except the crossing over. ಆ X shaped ಏನೋ ಒಂದು crossing over ಇರ್ತೈತಿ, that is nothing but a chiasmata. Then after diplotin, it is entered into the dikinesis. Dikinesis, it is the termination of the chiasmata. Termination of the chiasmata ಅಂದ್ರೆ ಏನ? ಎಲ್ಲಿ exchange ಆಗಲ್ಲ ಅದು ಏನಾಗ್ತೈತಿ ಅಂದಾಗ separate ಆಗ್ತೈತಿ, and the chromosomes are fully condensed. Chromosome ಇಲ್ಲಿ ಏನಾಗ್ತಾವು fully condensed ಆಗ್ತಾವು, fully formed ಆಗಿರ್ತಾವು. Then spindle fibers also form, and then ಇಲ್ಲಿ ಏನಾಗ್ತೈತಿ ಇದೆಲ್ಲ process ಮುಗಿಯೋಣ. Then after the dikinesis, prophase 1 ಆಗೋಣ. ಯಾವ ಬರ್ತೈತಿ ನಿಮ್ಮ ಮುಂದೆ? metaphase 1. After this you are going to go for the metaphase 1. Then after this is to go for metaphase 1. Metaphase 1. In metaphase 1 ನಲ್ಲಿ ನಾವು ಇಲಾಗಣ again the similar procedure. In metaphase 1, what is going to happen? In metaphase 1, bivalent chromosomes are come and join on a equator. Similar process. ಮುಂದಿನ ಏನಲ್ಲ in metaphase 1 again the similar. After this, the bivalent chromosomes are what? Come and join on a equator, and the microtubules are opposite poles. Spindle fibers are paired homologous chromosomes. As similar to the metaphase ಏನಿಲ್ಲ mitosis ದಾಗ ಅದೇ ರೀತಿ. After the metaphase, what is happened in a prophase 1? Sorry, anaphase 1. In anaphase 1, the chromosomes are going to separate in their centromeres. The homologous sister chromatids are separated, and again they are going to start pulling towards the opposite end. The homologous chromosome separates while the sister chromatids remain associated with their chromosomes, and they are start pulling towards the opposite pole. Afterwards, last one is to come, that is the telophase 1. And this is prophase 1, metaphase 1, anaphase 1, telophase 1. These are similar with the mitosis. This is very important. That is the prophase 1. That is longer and complex when compared to the prophase of a metaphase. And in telophase 1, what is going to happen? The similar process in the sense the nuclear membrane and nucleolus appears. In prophase nuclear membrane, nucleolus disappear. That is going to appear once again, and then it is followed by the cytokinesis. Ananತರ telophase ಏನಾಗೋದು cytokinesis ಆಗ್ತೈತಿ. Cytokinesis in the sense what? Division of cytoplasm. Then here we are going to form a 2 cells. ಅಲ್ಲಿ ಏನಾಗ್ತಾವು ಡಯರ್ 2 ಸೆಲ್ ಆಗ್ತಾವು. After that again it is entered into the what? We are going to go for the stage between the two meiotic division is called as a interkinesis, and is generally short lived. Ananತರ ಏನಾಗ್ತೈತಿ interkinesis ಆಗ್ತೈತಿ. That interkinesis it is between the meiosis 1 and meiosis 2. Interkinesis, it is the stage between the mitosis division and it is short lived. Then interkinesis is followed by the prophase 2 and much similar phases of the next. ಅದರ ನಂತರ ಏನಾಗ್ತೈತಿ ನೆಕ್ಸ್ಟ್ ನೀವು ಏನು ಮಾಡಬಹುದು meiosis 1 by prophase 1 ಆಯ್ತಾ prophase 2, then metaphase 2, anaphase 2, telophase 2. Those we are going to study next class. ಅವನೇನ್ ಮಾಡಬಹುದು ಅಂತಂದಾಗ ನಾವು ನೆಕ್ಸ್ಟ್ ಕ್ಲಾಸ್ ನ ಕಲಿಬಹುದು. ಆರ್ ಎಲ್ಸ್ ಇಲ್ಲಿ ಇಷ್ಟ ನೆನಪಿಟ್ಕೋರಿ, ಈ ಟೀಲೋಫೇಸ್ ಆಗನೇ ಏನಂತೀವಿ ಅಂತಂದಾಗ cytokinesis, and this cytokinesis it is going to form what I told you about the dyad cells. Then after that meiosis 1 and meiosis 2. Here we are going to get one thing, that is we are going to call that as a interkinesis. Interkinesis. What I told you? Interkinesis. Interkinesis. And then after this interkinesis, now we are studying prophase 1, metaphase 1, anaphase 1, telophase 1. After this interkinesis, you are going to go for prophase 2, metaphase 2, anaphase 2, and telophase 2. And these all are similar what we were studied in a mitosis. Mitosis like ಅಂತೀವಲ್ಲ ಅದೇ ರೀತಿ ಇಲ್ಲಿ ಏನಿರಬೇಕು ಅಂತಂದಾಗ ನ್ಯೂಟ್ರಲ್ ಡಿವಿಷನ್ ಕಂಟಿನ್ಯೂ ಆಗ್ತೈತಿ. and at the end after this telophase how many 4 cells we are going to get. ಆ ಒಂದು ಎತ್ತರದ after the what last, that is what we are going to call as a telophase 2, and telophase 2 are in cytokinesis. 4 cells are going to form, and those 4 cells are what n in number, in the sense those all are the diploid. Those all are in a means those all are diploid in nature, right? This is about what meiosis. Meiosis daga prophase 1, prophase 1 daga leptotin, zygotin, packaging, diplotin, dikinesis. In diplotin, leptotin, chromosomes are going to form. Zygotin, pairing occurs, we are going to call that as synapsis. In packaging, crossing over happens, that is the recombination, and it is going to form what? Recombination. And enzymes are involved, that is called as a recombinase. Then in diplotin, the separation of the chromosomes, and that is what we are going to call as a chiasmata. Dikinesis again is the complete termination of the chiasmata. Then after this dikinesis, cell means the division. Next division is metaphase 1, that is similar what we are studying in mitosis. Then metaphase 1 to anaphase 1 to telophase 1. After telophase 1, cytokinesis. Cyto means division of what? Cytoplasm. Then it is going to form a dyad cells, and in between the meiosis 1 meiosis 2 we are going to get the interkinesis, and after interkinesis again it is entered into the meiosis 2, that is the prophase 2, metaphase 2, anaphase 2, and telophase 2, and at the end you are going to get the 4 cells. This is about what meiosis. In meiosis, here we are studying regarding the prophase 1. In prophase 1, leptotin, zygotin, packaging, diplotin, and dikinesis. This is very important. If 5 marks question ಕೇಳ್ತಾರೋ 5 marks question ಕೇಳಿದಾಗ ಇಷ್ಟೆಲ್ಲ ಬರೀಬೇಕು. ಇದು ಅನಂತರ ಇವತ್ತು ನೀವೇನಪ್ಪ ಅಂತಂದಾಗ you go through book and write down the notes. Easy ಐತೆ. ಮೈಟೋಸಿಸ್ ಆಗಿ ವರ್ಷ ಅಲ್ಲ ಅದೇ ರೀತಿ ಬರೆಯಬೇಕು. But you are going to mention here bivalent chromosomes. Now after this, what we are going to study? We are going to study about the significance of meiosis. Significance of mitosis ಏನು ಕಲ್ತಿದ್ವಿ ನಾವು? Cell growth and division. It is due to the what we are going to call that one as a mitosis. Here, meiosis is the mechanism by which conversion of a specific chromosome number each species is achieved across the generation of sexual reproducing organism. The meiosis, it is very helpful for a sexually reproducing organism, and what we are going to go for, the results of the reduction chromosome number have and it increase. Important significance of meiosis is it is going to increase the genetic variability in the population of organism from one generation to the next. Variations are very important for the process of evolution. So, and here I am going to write here the significance. of what? Meiosis. Simple version. Meiosis significant ಏನು பார்த்தಂದಾಗ it increases the variability in the sense what? Variations. It increase the variations, genetic variations. Andre one generation to another generation genetic variations ಮಾಡ್ತಾವು, and this variability is helpful for evolution. This is helpful for evolution. We are going to discuss about the evolution when I am going to what in now Google use ಮಾಡ್ತೀವಿ, Google ಬಿಟ್ಟು ನಾವು evolution ಬಗ್ಗೆ ಡಿಸ್ಕಸ್ ಮಾಡಿ. Evolution in the sense it is a gradual change in organisms. Gradual change in organisms ಅಂತ ನಾವು ಕರೀತೀವಿ. Okay, and evolution ನಾವು ಕಲಿಬೇಕಾದ್ರೆ it is a broad what subject and ಒಂದು ತಕ್ಷಣ ಕಲಿಯಾಗಿಲ್ಲ. But evolution in the sense what? The gradual change take place in an organism and it is going to form a new organism or a new population. That is what evolution. ಇಲ್ಲಿ ಒಂದು ಸಿಂಪಲ್ ಆಗಿ ಹೇಳಬೇಕಂದ್ರೆ ನಾ the meiosis ಏನು ಮಾಡ್ತೀವಿ ಅಂದ್ರೆ meiosis it is increases the variability and it helps to the evolution. This is very important about the significance of meiosis, and this completes what? Meiosis. In meiosis it is a reductional division and daughter cells are having their chromosome half of its parental cell, and here the meiosis is take place in a meiosis 1 and meiosis 2, and in meiosis 1 we are going to study the prophase 1, metaphase 1, anaphase 1 and telophase 1, and a significant phase of the meiosis 1, that is the prophase 1, and that prophase 1 again followed by the different phases leptotin, cytokine, packaging, protein and diagnosis, and remaining whatever the metaphase 1, anaphase 1, telophase 1, then again the cytokinesis, prophase 2, metaphase 2, anaphase 2 and telophase 2 are similar like the mitosis. This is about what? Meiosis. And significance by meiosis, it is very significant in sexually reproducing organism. It is going to provide the variability in the organisms or a genetic combination from one generation to the another generation, and this meiosis is very important for the process of evolution. Okay, this is about the meiosis. And I am going to stop here, and this completes your chapter cell cycle and cell division. Okay, once again we are going to discuss regarding this in a Google Meet. Right? I will stop here. Thank you. I'll continue in the next class. Thank you.